Hello, everyone. I am Abir Ashra. I'm coming from Maynooth University, the Hamilton Institute. Today, I will be presenting to you thermodynamically favored DNA computation and storage, which we say it's robust, programmable, scalable, fast, and renewable. So I hope that I will convince you that it is these things. This project, or this work, was done under the DISCO project, which is one of the Digna portfolio funded by the EIC. Our partners were our program.dev and Telibet program is a French company, and Telibet is from Germany. And thank you very much for having me here and for bringing DNA computing to the DNA storage community. So to start, how we say what is thermodynamically favored computation is. In general, a classical or non-equilibrium, in a classical or non-equilibrium computer, the output is not the most thermodynamically favored, which means it is not the one with the least energy state. That means with time, it will either leak if it's a strand-displaced system or supernucleate or whatever, and then it will end up in an error state. However, in the opposite, if it's a thermodynamically favored, so the structure or your output should be the least energetically possible. So even if an error happens, it will correct itself eventually, and it will be the output state. This was originally inspired by origami. So origami, you might have seen it in a few talks before. Origami is done by having a long strand, as you see here in the video by Sean Douglas. This is M13. Long DNA strands, a single strand, and shorter, what we call them stables. And they utilize the complementarity sites of the DNA just to bring the scaffold together in specific parts, and with it, you can build different structures, either in 2D, like the first ones that were by Rudman here in 2006, or you can even do 3D stuff. Origami has been there for so long now as a technology and had been used in different applications, but the thing is it does not compute, and we were inspired to build something that is like the origami. That's the structure is the favored or very stable and very robust, but with computation. So today I'm presenting to you the Scaffolded DNA Computer, which is our work, or what we call SDC. And it's different from what Albert showed you yesterday, his SDC, so this is a different SDC. In SDC, we assume that we have a scaffold with a number of positions. On those positions, you can see for each position, you have some tiles that they are all allowed to bind to that position. Except for the first position, we allow only one tile, which we call it the anchor tile. So how it works, when a tile attaches to a position, a place in the scaffold, any position, either the previous or the following position has some tiles competing. So in this example, we have two tiles there. They both can bind to this side. However, you can see with the sides, the colors of the sides of the two tiles, only one of them is matching the tile on the scaffold. So the natural path for it is the correct tile will bind. So the two colors matching, you can imagine them as glues. So that's the correct binding. However, since the other tile is allowed to be there, so it can bind, but there will be a mismatch between the two tiles here, and because the other tile is already there in solution, so it can replace the previous or the wrong tile, and at the end of the computation, you will have everything matching, so that's your favored structure or the output. So from now on, imagine with me, because later on I'll be just showing you a bunch of squares, but imagine each square is a strand, is a DNA strand with 3 sides. So the 2 sides on the right and left, is where we encode the program and the data. And the bottom part is where it binds to the scaffold. So it's 3 sides. The bottom one is 24 bases, and they're all complementary to the scaffold. And the 2 sides on the left and right are 12 nucleotides, and they allow tiles or strands to bind to each other. So I'll show you the simplest computation we can do with this. Simplest in the mean of the computation itself, but for the SDC, I would say it's the hardest because you have the highest complexity possible. So this is bit copy, and by bit copy, it's just, you can imagine it like a wire transmitting a signal, either 0 or 1. So, and the first position, as I mentioned, it's only one tile. Here, we want to copy a 0, and then the following, we have 2 tiles per position. One of them is copying 0, and the other is copying 1. So if you put all of this in a test tube, heat it up so everything is melted, and start dropping your temperature, so what will happen that the tiles will start binding one by one, so randomly. So the first tile C, and then B, and then D, and then A comes, and it finds that the following is wrong, so it corrects the computation, and at the end you will be getting the correct answer, which is copying 0. So to do like actual computation, not bit copy, so let's program finite state machines. So how we do that, I mentioned at the beginning that we encode the data and the program on the left and the side. So this is where our program is. So for any finite state machine, we encode the state and the read symbols on the left, and the result of your computation, which is the following state depending on the read, is encoded on the right, and then we encode the next read along with it. So we use that to encode 3 bits per side. So this is an example of addition. So addition, this is a finite state machine that the states are the carry is either 0 or 1, and the transition is the inputs that you have. So adding 2 numbers, so you add the first 2 bits, so these are the transition that you are getting, and the carry of the next carry is the next state, and the previous carry is the state that you are in. So for each position, we have 2 tiles. One of them is assuming that the previous carry is 0, and the other is assuming that the previous carry is 1. They don't know what they will get. So this is a full computation of 10 plus 3 equals 13 in binary, encoding the data as mentioned above, and then the result is 1, 0, 1, 1 in binary, and this is what we get experimentally. So when you heat, so these are, as I mentioned, imagine them as DNA strands. This is the scaffold, the long scaffold, and these are just the short strands. So this is the result in annealing. So 1, 1, 0, 1, the red one is high, and then the green one is high, and then the orange one is low. So that's the orange 0, that's orange, that third position, so that's low. And then the last one is, the blue one is also high. So that's 13. And how we report that, we report them in separate test tubes. So we have 4 test tubes, each of them is reporting at each position. With the same principle, we did, so I told you it's programmable, so we programmed 10 different programs, addition, and these other 9 programs. Each of them has different features. I'm happy to talk to anyone who's interested in knowing more about them, but I'll just highlight, for example, this one in the middle. It's not only binary now, we can detect more than 2 levels of signal. So we have high signal, we have low signal, and we have a signal in between. So that made us be able to report or work on threads. So 3 digits system. This counter and rule 110, for example, they have 8 tiles competing for position. So that's higher complexity. We also managed to do NFA, parity checking, even balanced brackets. So like 10 different programs. I also told you it's fast. So the previous results I showed where the annealing was 3 hours, so over dropping the temperature 1 Celsius per minute. However, you can get your result as fast as 1 minute. So heat everything in the tube to 80 Celsius, and then drop it quickly, and within 1 minute, you can get your result if it's 0 or 1. So this is also 10 plus 3 equals 13 that I showed you a bit ago, and it's the same result. One nice thing about this slide is that these 2 programs were in a plate that I initially mixed in 2023. And then we submitted the paper, and then we got the review. And before replying to the reviewers, I found the plate in the fridge. I just took it out. That was 1.5 years later. Most of the liquid was evaporated. I eyeballed, like, added the liquid to the 96 Wells plate and just put it in the qPCR, and I got this result. So this result is coming from after mixing the plate 1.5 years afterwards. So it is, we think that's why we think it's also robust. I told you it's renewable. So it is renewable by nature, because if you have this instance in the tube and you do the annealing, you will get your structure. But if you heat it up again and cool it down, so you will get the same structure, so you can reset your system multiple times. But resetting is not enough. We want to reset and compute a different thing. So we can do that. How we can do that? So if you have the system and then you get your first computation, so just have the input and get your result. So heat it up again, but in that, while you're heating up, block the anchor or the first tile. So block the first tile, and then we add another tile, so we get a different computation. You can do that multiple times. And that's what made us actually think of applying to the Pathfinder challenge, because we found that our system can do dynamic storage. So you compute, store, and then compute, store, and so on. So I'll show you an actual result. That's bitcopy. That's the first that we tried it with, and I'll show you others. So in bitcopy, these are the actual strands on the scaffold copying 1. So here we report with the fluorophore quencher pair. So the quencher is on the scaffold. So the fluorophore is on the scaffold, and the quencher is far away. So that's how we get the high signal. So we have a 1 here. That's copying 1. And then we block it, and by blocking it is just to add the complement of the anchor here. So when you're heating that, when you're heating up and cooling down, what will happen is that the anchor the previous anchor will bind to its blocker very early, so it will be blocked, and that would allow the new input to work on the structure, and you will be copying 0. So that's the low signal. And then you can do it again and again and again, and we did that up to, like, I was tired by the end of this experiment, so that's 25 times. But still, you can do it more. So at the end, we have this high signal, and it's still differentiable from the 0, so you can do it more. We also did addition. So by only changing the first 2 bits in each of the numbers, you can get a different computation. So this is the same program, but renewed 8 times with different computation every time. And this one is also, it started with adding 12 plus 3 equals 15 and ends with 12 plus 0 equals 12, and then reset 12 equals plus 3 equals 13 again. This one is one of my favorite because it's the counter that I told you before, that you have 8 strands and they are all competing over the same position. But every time we change the anchor, so instead of counting 0, 1, 1, 2, 2, 3, 3, 4, this is the first computation here. So the one that's low, it is the correct counting. So you block this one and you add the following, which should be 1, 2. So it will be taking this 2, 3, and then 3, 4, and 4, 5, and that's the second computation, and so on. This is also renewed and reused 24 times. Last but not least, we say that SDC is also scalable. So until the previous slide, what I was showing you was done on a scaffold that the sequence was outsourced from the M13, yes, but it was Alzheimer 120 nucleotide single strand that we ordered from IDT. But to scale more, so we wanted to do, for example, 25 position, because each position is 24 bases, so the total will be 600 and something. So that to get that from IDT, it was very expensive, and the yield would be too low. So we thought, okay, let's just move to M13. So starting from here, we are running the computation over the M13, so you can imagine like the 7,000 single strand is there in solution, and we still manage to compute. This is an example of adding these 2 big numbers with millions to make two big numbers in binary. These are the 2 numbers in binary. This is from the least significant bit to most significant bit, and this is the result. So I'm just showing you the finishing level. So that's high, so that's 1, 1, and then 0, 1, 0, 0, 0, 1, 1, and so on. However, this is good and impressive, but if you look at the lighter color triangles, they are shorter anneals. So to achieve the good result, we needed to do longer anneals, which are the darker triangles. So the darker triangles are done on 24 hours. So the annealing protocol was 24 hours. And we believe this happened because there might have been some kinetic traps due to some constraints that were hard for us because we were working directly on the M13, so we don't have control over the sequence of the M13. So some positions, for example, here were very stronger than others. This is just the strength, the MFE of each of the of each of the domains. So some of them were much stronger. So if you get an error in that position, so it might propagate. So to solve that, there was another thing that we also looked at, which is what we call sync and no sync. It depends on the time if I can talk about it, but I can talk about it more later. But you might get it when I get to the isoenergetic thing. So in the previous one, we had structures like this. If you are reporting high, for example, you would get a structure, and if you are reporting low, you will get another structure, but their delta G is different. One of them is stronger than the other. So we thought of doing the isoenergetic, or adding the isoenergetic feature. So if you start with a 0, so you will get the sequence for 1, and then back to the sequence of 0, back to the sequence of 1, and so on. And if you start off with a 1, you will change to 0, and so on. So using two sequences, interchanging between them to make the two result structures are kind of equivalent in their free energy. And that's what we call isoenergetic bit copy. And this is the result of the isoenergetic bit copy, which is much better than if we do the regular bit copy. So this is 14-hour protocol. In each of the positions, I'm showing you copying 0 on the bottom and copying 1 on the top. So we managed to get good separation until like the 25th position. So to summarize, scaffolded or SDC is robust. As I showed you, the one result that was 1.5 years old. It also ran very well. One thing, it doesn't need a special anneal or purification or anything. Our strands are ordered unpurified. We just mix them, and we don't need to measure the concentrations very carefully. So that's another thing. It's also programmable. We ran 10 different programs. It's fast. In terms of the length, so in the 4 positions, it's very fast. We can get it in 1 minute. In the longer ones, we need a bit longer, so we can do it in like a day or something. You can get the good 75-bit computation. It's renewable. It is also scalable, depending on the sequence, of course. There's more work to be done on the scalability. So what we are looking forward to do next, we want to scale it up more, up to 1 kilobit, which is computing and storage together. We also want to automate the renewal process to eliminate the addition of the strands. We would like to do random access. So far in renewability, what we do is just we can change only the first strand, which is the anchor. We want to actually go to any other position and just to change it if we want. So that would help us to get random access. We are also looking forward when we do the scale up. So the current way of how we report will not be helpful, will not be good. So we are thinking of high throughput readout like sequencing, for example. And also we would like to extend it instead. So you saw now we are working on the M13 and just 1-dimensional. We want to do it 2-dimensional on the origami, so you can branch on different directions. Finally, I would like to thank everyone here in the conference very much for inviting us and our partners, Telibet and Bregma.dev, the DESCO project and all the members. Most of you know Damien. He's the head, the lab head, and the other colleagues, Tristan Stiron. He is my joint co-author in that work, and everyone else. Also, another thing, on September, I'll be starting my own grant. So I'm hiring a PhD and a postdoc. So I hope it's okay to announce that here. So if you are or anyone if you know anyone else who's interested, please reach to me. Last thing is I invite you all to know about DNA 31. So that's another conference. It's a computing conference. It's most focused on computing, but it's also welcoming data storage, of course. It will be this year here in Lyon, in France, in August. So we will have lots of our research presented there, so you can come and talk to us. We would like to see you there. Thank you very much.
Do we have questions? Yes.
Thank you so much for this really fascinating, exciting, amazing work. I am really excited. Yeah, it was very great work. My question is, I'm a chemist. I am a bit far from 0 and 1 and just converting this. My question was that about how did you do the calculation? Okay, let's say you did 13 plus 9, I guess. I can understand you turned 13 into one sequence and 9 in another sequence. How did you make the plus?
So it's not the 13 in a sequence. It's each bit or each computation. So this. Okay, so each 2 bits are encoded on the left. So you can see here 0, 1, 0 plus 1 are encoded here, and the previous carry is encoded here. So this is in binary is 0, 0, 1. So we have in each position we have 8 different sequences that you can choose from. So we have a bool. Let me go to. I have some extra slides, so maybe I would need to. Yeah, that's better because they are hidden. Is it this one? Yeah. So we designed this bool initially for the 4 positions. We have 395 different strands because in each position you have 8 different sequences on the left and 8 sequences on the right. So the total per position is 64 strands. And then this is, for example, that's a different example than the addition, which is parity, doing the same encoding method. So this to hold, we call it 0, 1, 0, which is 2. So just go get the compute domain that has the sequence that's related to it. Am I answering your question?
Okay, but I still don't know how can you do the plus part. Okay.
It's hard-coded, if that's your question. So the plus part, it's that's another. I don't think I'll be able to represent this, but it's hard-coded. So you choose the strands from the pool. So we know that here we need compute domain 2, here we need compute domain 3. So you choose the strand that has compute domain or the sequence that's equivalent to compute domain 2, and then A, and then 3. So the strand is called 2A3, for example.
I see. I didn't get it, but later I will ask you.
Yeah, sure.
Thank you.
Okay, yeah, I'm happy to clarify.
Other question?
Yeah, I would have one. Thank you very much for this really interesting talk. Remind me, or if I didn't catch it, how do you read out the data? You do qPCR, you said, and then sequencing, or what?
We do qPCR, not sequencing, and this is how we do the data. So for example, here, this is the 4 positions. We always use an extended position, so one more position to place a fluorophore. And in the position before it, you have 2 strands competing. So we hard code one of them, we add a quencher to it, which is this one, and the other does not have the quencher. So if the one selected, that result is high or 1, so that's the one that will be on the scaffold, and the other will be just flowing in solution, so you will have a high signal. And to do that, as I mentioned previously, we do it in 4 different test tubes. Currently, that's how we do it, because you cannot do the 4 of them in the same test tube. You cannot put 4 fluorophore and detect the 4 signals at the same time.
Also, only for the last position, you have this quencher.
Yeah,
not for the
the
ones
fluorophore.
before. The fluor 4, sorry.
Yeah.
Fluor 4. Okay, not for the ones before.
But for the ones before, we have them in different test tubes. So in a test tube, you have the computation here, and your fluorophore is on this position. And in the third test tube, you have your computation up to here, and the fluorophore on this position, and so on. This is just showing you the last position.
So it's a lot of different reactions, test tubes,
yeah,
and then
different test tubes.
putting them into one.
qPCR.
Oh, okay, okay.
Yeah.
Thank you. Okay,
Sure.
thank you.
Last one, anyone? Okay, thank you very much.
Thank you, David.